My name is Leonie. I am currently a group leader at the Department of Plant Sciences, working on beneficial symbiosis between plants and microbes. But before I started my group, I was actually a postdoc in the department in Luginbuehl's lab, working on carbon assimilation. And both of these topics that I worked on and that I currently work on have to do with carbon. And so I think during the talk, hopefully I'll get a chance to explain a bit more why I think it is really important to look at carbon. So what I want to do today is to tell you about carbon and tell you about the importance of carbon for crop yield and food security. And I will talk specifically about carbon assimilation and allocation at different scales, all the way from single cells to plant microbial systems. And so I will actually present in the first part of my talk some of the work that I did as a postdoc in Julian's lab. This was a collaboration with the Salk Institute in San Diego, in particular with Joseph Swift, who is a postdoc in Joe Ecker's lab. And in this project, we looked at carbon fixation, so the first step of the path of carbon in a plant in leaves. And so we only just 2 days ago published the paper, so it's now online and accessible to read, but I wanted to show you some of the highlights of that work. But then I also want to spend just a couple of minutes at the end of the presentation to tell you a little bit about the research interests of my group. And we're still very interested in carbon, but instead of focusing just on carbon fixation, and you can see them all sitting here, instead of focusing just on carbon fixation, we look at how fixed carbon is used in a plant, in particular in beneficial plant microbe interactions. So hopefully there will be something interesting for many different people who are listening, even if maybe you don't have a background in plant molecular biology. So let's start with the very first step of carbon fixation. The efficiency with which plants can fix carbon in the leaves is actually one of the key factors that limit maximum crop yield. And you can see here a picture of plants. All of these plants use a particular way to photosynthesize that we call the C3 photosynthetic pathway. And this includes important crops such as rice, but also wheat, potato, and tomato. And all of these plants use C3 photosynthesis to fix carbon in their leaves. The problem with C3 photosynthesis is that it isn't particularly efficient, particularly at high temperatures and in dry environments. And so about 30 million years ago, some plants evolved a more efficient way to photosynthesize, to fix carbon, which we call C4 photosynthesis. And these include plants like sorghum that you can see here, but also maize and teff, for example, but also sugarcane. And these plants, because they use a more efficient way to photosynthesize, they show increased carbon fixation efficiency, but also increased nitrogen use and water use efficiency. And that actually can increase their yields by up to 50% compared to the most productive C3 crops. And so as a result of that, these crops are some of the most productive crops on Earth. So what makes C4 photosynthesis so much more efficient than C3 photosynthesis? To understand that, we have to look at the different cell types in the leaves and their role in carbon fixation. So what you can see here are cross-sections of a rice leaf on the left, rice is a C3 plant, and a sorghum leaf on the right. Sorghum is a C4 plant. And these are electron microscopy images, and you can see the different cells making up the leaf here. And what we typically see in a C3 leaf, like rice, is that photosynthesis primarily takes place in the mesophyll cells, indicated in green here. So the mesophyll cells are fully packed with chloroplasts, and you can see these little blobs, which are the chloroplasts. And so photosynthesis takes place primarily in this cell type. And compared to the mesophyll cells, the bundle sheath cells, which surround the vein in the middle here, they contain much smaller chloroplasts, and they're generally less photosynthetic. So in a C3 leaf, photosynthesis takes place in these mesophyll cells. This is where the primary enzyme for carbon fixation, rubisco, is active. and it fixes carbon dioxide here into C3 sugars. Now, Rubisco is not always very specific. And instead of using carbon dioxide, Rubisco sometimes uses oxygen as a substrate. And that's problematic because during this reaction, a toxic byproduct called phosphoglycolate is being produced. And so this phosphoglycolate then has to be recycled by the plant to remove it from metabolism in a process that we call photorespiration. And this photorespiration not only is energy consuming, but it actually releases carbon dioxide. So it's the opposite of what we want during carbon fixation. And this oxygenation reaction, this unspecific reaction that Rubisco carries out, happens more frequently when the temperatures are higher. And so that's why C3 plants are actually not particularly efficient in hot environments. Now, C4 plants have evolved a way to basically almost completely suppress photorespiration. And they do that by partitioning the photosynthetic reactions between two different cell types. They partition it between the mesophyll cells and the bundle sheath cells. So what happens here is that we have the first step of carbon fixation in the mesophyll cells, and it's now actually not carried out by Rubisco, but instead by an enzyme called PEP carboxylase. And this PEP carboxylase produces C4 acids, and that's where the name C4 photosynthesis comes from. These C4 acids are then shuttled, transported to the neighboring bundle sheath cells, and they're then decarboxylated around Rubisco, which now is present in the bundle sheath here. And you can then see that this will increase the concentration of carbon dioxide around Rubisco, which actually means that the oxygenation reaction is almost entirely suppressed. And Rubisco then creates these C3 sugars with this carbon dioxide that can then fuel metabolism in a plant. And so by separating these reactions and limiting carbon fixation by Rubisco to the bundle sheath and concentrating carbon dioxide around Rubisco, photorespiration is suppressed. And that makes these plants particularly good at growing in hot and also in dry environments because it simultaneously increases water use efficiency. Now, the photosynthetic pathway, did you want to say something, Abigail, or?
No.
Okay, good. Sorry. The C4 photosynthetic pathway actually evolved many times independently from the ancestral C3 pathway. In fact, more than 60 times independently. And you can see here a tree of the different plant lineages. And in blue, we have the origins of C4 photosynthesis. And you can see that this pathway evolved in many different plant lineages independently. And so what we know is that, in fact, the C3 plants, they already contain all of the genes that are necessary for C4 photosynthesis. The only difference, or one of the main differences, is how these genes are regulated to achieve a C4 cycle. And so that then led to this idea that maybe we could use that, and because it happens so many times in nature already, maybe we could use a similar strategy to try and introduce the C4 pathway into C3 crops. So, for example, can we engineer rice such that it now operates the C4 cycle, and so make these C3 crops more resilient to climate change and better able to grow in hot and dry climates? And this is something that a lot of different research groups are working on currently across the world. But to be able to achieve that, we have to understand how exactly plants evolved C4 photosynthesis. And one of the steps that is always common during the evolution of C4 photosynthesis is that plants had to change the way in which they regulate photosynthesis genes. So if we look at these leaf cross sections again, what we can see on the left in rice is that Rubisco is most highly expressed and present in the mesophyll. But to operate the C4 cycle, Rubisco has to then be expressed specifically in the bundle sheath. And so there has been a recruitment of photosynthesis genes into the bundle sheath. And in fact, you can see that the bundle sheath in a C4 plant now contains really large chloroplasts. And so there's a general activation of photosynthesis in the bundle sheath. And other enzymes had to change their expression as well in the leaf. And this is what we try to understand in this collaborative project with the Salk Institute. In particular, how did photosynthesis gene expression change in these different cell types to allow the evolution of the C4 pathway? And so we did this work in rice and in sorghum to compare what happens in a C3 and in a C4 leaf. And I want to just show you two of the main findings from this work and talk a little bit about the significance of these findings. So one of the key regulators of photosynthesis gene expression is light, and that's true for both C3 and C4 plants. So if we grow these plants like rice and sorghum in the dark, and they have this yellow appearance, a bit like white asparagus that hasn't seen light. They don't photosynthesize in the dark, but as soon as we expose these seedlings to light, they start greening up and they start photosynthesizing. And that's because the genes involved in photosynthesis are activated by light. Now, because this is true for both C3 and C4 plants, the real question here is then, how are photosynthesis genes switched on, for example, only in the mesophyll in rice, but then instead in sorghum in the bundle sheath? So we decided to use this system looking at dark-grown and light-grown plants and try and understand how photosynthesis gene expression is regulated in each cell type to really get to these differences. And for that, we used a technique called single-cell sequencing. So we quantified gene expression at single-cell resolution. And just briefly, what we did was we froze the plant tissues and we then extracted the nuclei. We didn't extract each individual cell, but instead focused on the nucleus of each cell as a representative of each cell. And by staining these nuclei with a fluorescent dye and then using fluorescence-activated sorting of these nuclei. And then we used single nucleus sequencing of RNA and DNA using different techniques. And I'm happy to talk about these in more detail if anyone is interested after the talk. So this then allowed us to look at gene expression in each nucleus and so in each cell individually and try to figure out what the differences are between rice and sorghum. And so we sequenced more than 100,000 nuclei for both of these species in the shoot. And this is how the data looks like. So what we do is we create something that we call a UMAP. And each dot that you can see on this graph is the transcriptome of a nucleus, so the gene expression profile of an individual nucleus. And what you can see is that the closer together two nuclei are here, for example, the more similar their gene expression profile is. And so the idea is then that, for example, if we have a particular cluster, these are clusters of nuclei that share common gene expression patterns. And we then can use that information to figure out what these nuclei, which cell type these nuclei belong to. So we can annotate the clusters to these different cell types and figure out where is each nucleus coming from, what cell type did it belong to. And for that, we can use certain genes that we know should be expressed in each cell type. So in this way, we were able to create gene expression atlases for rice and sorghum shoots. And we could, for example, look specifically at the mesophyll nuclei, which are these nuclei in clusters 1 and 2 in rice, and the bundle sheath nuclei in cluster 8 here. And in sorghum, again, we particularly focused on the bundle sheath cluster here and the mesophyll cluster here. So that allowed us to really look at each cell type individually. So the first question that we wanted to ask was, how different is gene expression in a mature C4 bundle sheath? How much has gene expression actually changed? This was not something that we were able to do before. And so we looked at the bundle sheath in rice and the bundle sheath in sorghum and looked at the functions of the genes that are specifically expressed in these cell types. And we found that in a C3 plant, these genes are mostly involved in transporting solutes, transporting water in the plant. In a C4 plant, however, we found that these genes that are associated with the bundle sheath are actually involved in small molecule metabolism, organic acid metabolism, and the generation of precursor metabolites and energy. And that reflects the role of bundle sheath cells in C4 photosynthesis. So already from looking at the functions of these genes, we saw that actually the functions are really different. And we then looked at the overlap of gene expression in each cell type. So here you can see an extent of overlap of gene expression of the most specific genes in each cell type in sorghum and in rice. The darker the color here, the more overlap we get, the more conserved gene expression is. And we can see that some cell types are actually really conserved. The same genes are expressed in these cells in sorghum and rice. You can see that for mesophyll here, but also for the xylem. But then surprisingly, the bundle sheath is actually the least conserved cell type, meaning that it's completely different sets of genes that are expressed in a bundle sheath in a C3 plant compared to a C4 plant. And we can see that here as well. If we look at the bundle sheath genes that are expressed in sorghum here in blue, we can actually then look at where they are expressed in rice, in which cell type. And you can see that only very few genes are expressed in a bundle sheath of sorghum and rice. But actually, most genes that are expressed in a bundle sheath in sorghum actually are expressed in a mesophyll in rice. And that includes a lot of photosynthesis genes. So there's been a huge rearrangement of gene expression for the bundle sheath to allow C4 photosynthesis, and in particular, a recruitment of mesophyll-specific genes in a C3 state to the bundle sheath in a C4 plant. The second question that we looked at, and the second result that I want to share is how are photosynthesis genes then recruited? So how are these genes regulated so that they now are expressed specifically in a bundle sheath, such as Rubisco, for example? What are the gene regulatory mechanisms that have allowed this change in gene expression? And when we talk about gene regulation, often we talk about transcription factors and cis elements. So if we look at DNA and we look at a protein coding sequence here, we can activate the expression of this protein coding sequence. And often the regulation happens a little bit upstream of this protein coding sequence. And this is where transcription factors can bind. These are proteins that can either activate or repress gene expression. And so the more transcription factor we have, for example, the more activation we can get of gene expression. But these transcription factors don't just randomly bind any DNA sequence, but in fact, they're quite sequence specific. So they will bind, for example, a sequence that is GGP, AGGP. And this is what we call a cis regulatory element. So if we think about changing gene expression, we have two ways in which we can do that. Either we can change the amount of transcription factor that is present in a cell to activate gene expression more or less, or we can in fact change the DNA sequence that is bound by transcription factors in this gene regulatory sequence of the DNA here. And if we change these cis elements because we have sequence specificity, it means that a new cis element could recruit a different transcription factor that might already be present in the cell. So we looked at these possibilities and looked specifically at the cis elements that are present or accessible and enriched in each cell type of rice and sorghum. And then we compared which cis elements are active in each cell type. And when we do that, similar to before, we compare these cell types, we can see that actually there's quite a good conservation between rice and sorghum. So for example, in the mesophyll, we see this particular cis element being enriched in the mesophyll-specific genes in both species. And this sequence is bound by MIB-like transcription factors. We also see that, in fact, in a bundle sheath, we see enrichment of AAAG, of this particular sequence, which is bound by DOF transcription factors. And actually, that was really surprising to see that that's enriched in both sorghum, but also rice. So if we look back to the genes that are expressed in the bundle sheath, we found that it's very different sets of genes that are expressed in a bundle sheath in a C4 plant compared to a C3 plant. But from this result here, it looks like, in fact, the way that these genes are regulated in the bundle sheath or activated in the bundle sheath in both species goes through these DOF binding sites. And so the way that these genes are regulated, even though the genes themselves are different, the regulation is actually the same. And we were particularly interested in genes that swap expression from the C3 mesophyll to the C4 bundle sheath. So these are genes in rice that are more highly expressed here in the mesophyll, and that includes a lot of photosynthesis genes here. But then the same genes in sorghum, the exact same copies, are actually more strongly expressed in the bundle sheath. And if we look at the cis elements in these genes, we find that as expected in rice, we have these MIB-like transcription factors that bind to these cis elements and activate gene expression in the mesophyll. So this is what we saw before as well. But now the same genes here, actually in sorghum, start being enriched in the DOF binding site. So we again see AAAG coming up in these DNA sequences, which actually means that this is what we now think is an evolution in cis. So actually the DNA sequences themselves changed during evolution of C4 photosynthesis. And we also find that the DOF transcription factors themselves, and when we look at where they are present in the leaf, we find that they're actually mostly present in a bundle sheath in both species. So they don't seem to have changed. You can see that most of these DOF transcription factors, the proteins themselves, are expressed in the bundle sheath of rice, but they're also expressed in the bundle sheath of sorghum. So the transcription factors themselves have not changed, but the DNA sequences in the genes that are regulated by these transcription factors seem to have changed. And so going back to this model, at least in the genes that we looked at, we now think that this hypothesis too was true, that in fact these genes change the sequence here so that they now can recruit the DOF transcription factors that are already present in the bundle sheath. And just to summarize this part of the talk and end with a model that we now think explains at least some of the mechanisms for the evolution of C4 photosynthesis. We think that in an ancestral C3 leaf, what happens is that photosynthesis genes are active, particularly in the mesophyll cells, because they contain these mesophyll cis elements like the MIB-related transcription factor binding sites, whereas the photosynthesis genes, because they only contain these mesophyll motifs, they can't actually be activated by the DOF transcription factors. And instead, these DOF transcription factors activate bundle sheath specific genes in a C3 plant, which are other genes like these genes that we saw are responsible for the transport of solutes and water in C3 rice, for example. Now, during the evolution of C4 photosynthesis, what we now think happened is that these photosynthesis genes changed the sequence in this area of the gene here to now contain these DOF motifs, the DOF binding sites. And because the DOF transcription factors are already present in the bundle sheath here, they can now bind to these photosynthesis genes and activate their expression in the bundle sheath. And so we think that what was necessary here was the recruitment of these ancestral bundle sheath specific motifs, the DOF motifs, to allow recruiting of these photosynthesis genes into the bundle sheath. And this now could be a potential route for introducing the C4 pathway into C3 crops. What it means is that what we can try to do is use methods like, for example, CRISPR to change the DNA sequence of these genes and to try and get them activated in the bundle sheath in, for example, rice. But of course, this is just one mechanism, and we know that there's other mechanisms as well that have to be considered for potentially engineering a C4 pathway into C3 crops. But we hope that by doing that, eventually we'll be able to make plants that actually are much more resilient to climate change, especially important crops such as rice. Now, I've talked a lot about this first step of the path of carbon in a plant, carbon fixation. And in the last couple of minutes, I just want to also talk a little bit about what happens to fixed carbon once it's been fixed by the plant. I think that I mentioned initially that carbon fixation is a key factor that limits maximum crop yield. And that is definitely true, but I think it's equally important to consider how the plant uses this fixed carbon. because in fact, more than 80% of fixed carbon is exported from these source leaves where carbon fixation takes place to different sink tissues. So for example, a lot of the fixed carbon is actually transported to the root to support root growth and development. And there's a lot of carbon that goes into seeds during reproduction, and this of course is important for crop yield. But there's also a lot of fixed carbon that is transferred or is allocated to beneficial microbes. And the most widespread of these symbioses is the arbuscular mycorrhizal symbiosis. And in fact, most land plants, including crops such as rice and maize and sorghum, can form this symbiosis with arbuscular mycorrhizal fungi in the soil. And what happens here is that the spores in the soil, you can see these spores here, they germinate. These fungal hyphae that you can see here, the really thin hyphae, they start branching and they grow towards the root, which you can see here. And then they in fact colonize the surface of the root, but also enter the root cell and form these feeding structures in the root cortex, which we call arbuscules. These are very finely branched fungal hyphae that enter the root cells. And this allows the fungus to in fact get access to carbon from the plant. So this is where a plant then feeds the fungus with fixed carbon. And this is really important for these arbuscular mycorrhizal fungi, because without the plant and without this fixed carbon, they can't survive in the soil. They can't reproduce and complete their life cycle. They can only survive in the form of spores. Now, this carbon transfer not only is really important for the fungus to survive, but actually up to 30% of fixed carbon can be allocated to these mycorrhizal fungi in roots. And so it's a huge proportion of carbon that goes to these beneficial fungi. And at a global scale, it's really important as well. So this is a paper that was published about a year ago that looked at carbon transfer to these mycorrhizal fungi, not just arbuscular mycorrhiza, but also other mycorrhizal fungi that plants associate with. And these researchers estimated that more than 3.5 gigatons of carbon every year are allocated to mycorrhizal fungi in the soil. And this corresponds to more than a third of the current annual carbon dioxide emissions from fossil fuels. So it's a huge amount of carbon that is drawn down from the atmosphere into the plant and then allocated to these mycorrhizal fungi in the soil. And so that carbon transfer also plays a huge role in the global carbon cycle. Now, the reason why these plants provide so much carbon to these fungi is because they in fact trade the carbon for mineral nutrients. So the fungus is able to take up mineral nutrients from the soil much more efficiently than the plant root would be. These fungi have access to phosphate and organic nitrogen that they can transport from the soil to the roots and then release into the root cells in cells that contain these arbuscules. And that drastically improves nutrient uptake. And I've mentioned that carbon fixation is really important for crop yield, but of course we know that nutrient uptake also is extremely important. Here we can see rice plants that are grown with phosphate and without phosphate, and we can see a huge difference. And in fact, this arbuscular mycorrhizal symbiosis can actually improve crop yields by up to 30% by increasing a nutrient uptake. So here you can see an example of a maize plant on the right that can form a symbiosis with arbuscular mycorrhiza, and a plant that can't form this symbiosis. And you can see a huge difference in growth on these nutrient-poor soils. And that's predominantly because these fungi are so good at taking up these nutrients and provide these to the plant. And so overall, this symbiosis is an exchange of nutrients where fixed carbon through photosynthesis is exchanged for the nutrients. And what my group now looks at is how this nutrient exchange is regulated. In particular, what we want to understand is the role of carbon transfer in this symbiosis. At the moment, we have a very limited understanding of how carbon transfer is regulated by the plant or by environmental conditions. And so what we want to look at is, can we actually optimize this symbiotic nutrient exchange in crops by manipulating this carbon transfer? So one of the ideas that we're working on is to try and increase carbon allocation from the plant to the fungus. If we can feed the fungus with more carbon, this might increase fungal growth in the soil. It might support the hyphal network in the soil, which in turn could lead to better nutrient uptake through the fungus and more nutrient provision to the plant. And in turn, of course, that could then increase plant growth and yield. And so we're trying to figure out what the role of carbon is in this nutrient exchange and how we could potentially optimize that in crops such as rice. With the aim, of course, that if we manage to do that, if we can enhance this symbiosis in the field, we could reduce the need for synthetic fertilizers that are damaging for the environment and are expensive. But at the same time, this could be a possible route to sequestering more carbon in the soil and increasing soil fertility as well. And with that, I want to end. I just want to acknowledge the people that have been involved in the work again, in particular, Julian Hibbert, but also Joseph, who I've been collaborating with very closely over the last 5 years for the work in rice and sorghum. But then also all of my group members who are now doing amazing work to try and understand the arbuscular mycorrhizal symbiosis and the role of carbon in this symbiosis.